Hey all, so this video is the HL version of the blood system, so B3.2, and we're thinking about what adaptations facilitate transport of fluids in animals. We're looking at page 284 to 89, and again, we're still thinking about form and function. So structurally, how are these things designed or evolved to facilitate their function? So last video, we saw the heart structure, and knowing the parts of the heart and labeling them and identifying their function is an HL aspect. But our hearts are designed to move blood at high pressure. And part of that is because the ventricles are strong muscles. The atrium fill the ventricles before the ventricles pump. The valves prevent backflow. Cardiac muscle can simultaneously coordinatedly contract. And so that allows all the heart muscles to be working in sync. The pacemaker, the SA node, which is in the right atrium, signals the right atrium to contract by depolarizing first, and that's going to control the heart rate. And so it depolarizes, meaning it sends an electrical signal before the AV node, which is further into the right atrium, atrioventricular node, before it will depolarize and signal to the ventricles to contract. And then there's the septum, that's this big muscular divider here that's going to keep high and low oxygen blood separate from each other. Coronary arteries carry oxygen to the heart and glucose to the heart also, as well as dropping off carbon dioxide for removal. So heart muscle is structurally a little bit different than striated muscle, which makes up our muscle fibers in like our muscles or something. But in order for any muscle to contract, it must have a pulling force applied to it. And this is true for it to lengthen also. And so if we were talking about a bicep, in order for it to contract, my bicep contracts, but then I need an opposite muscle to lengthen my bicep, and that's going to be my tricep. And so these are called antagonistic pairs. We have them all throughout our body. And striated muscle, which the word striated means striped, and that's because striated muscles, like the muscles in your arms, have alternating dark and light bands, which we will talk about more when we look at muscle tissue. But that's the stripiness of the striated muscle. And so striated muscle, that's your bicep, tricep, quadricep, are attached to bones and move the body. They're long tubes of muscle fibers, which are called myofibrils. And they have many nuclei, and all those fibers are arranged in parallel. And those myofibrils have alternating dark and light bands. Cardiac muscle structurally is a little bit different. It looks somewhat similar to striated muscle, but the cells are much shorter. They only have one nuclei, and they have branching happening with a connection point called an intercalcated disc. And this intercalcated disc allows for signaling to happen simultaneously and quickly throughout the heart. And so this structure here, the intercalcated disc and this branching, allows for cardiac muscle to contract altogether. When we are talking about the heart, we do need to be able to interpret stages of the cardiac cycle. So looking at a diagram like this, we need to be looking at what's going on with the volume of blood in the heart, what's going on with the pressure in the heart. And so blood pressure and volume are going to change through different stages of the cardiac cycle, and that's due to muscle contraction and opening and closing of valves. The highest pressure and lowest volume is going to be when the ventricles contract. And so at this point here is when the ventricles contract. So you can see the pressure gets really high, but the volume gets really low. And the lowest pressure and the highest volume is going to be when the atrium are filling. So at this point here, the pressure is quite low and the volume is quite high. Additionally, if we're looking at an pardon me. If you're looking at an electrocardiogram, this measures the electrical signals in your heart. And so what you have a P wave, a QRS wave, and a T wave. And so the P wave is measuring when your atrium contracts. The QRS wave is when your ventricles contract, and then the T wave is when these semilunar valves close. And you can also see here that this is coordinated with the heart sounds. Heart has two sounds. It makes a like boom, boom sound. And what's going on there is these are the first valves closing. Those are the AV valves just before the ventricles contract. And then this is the semilunar valves right here after the blood is all squirted out through the arteries. Electrical signals causing contraction in the heart can be read with this electrocardiogram. And actually the size of the QRS wave, the distance between the P and the QRS wave, the distance, all of these things can tell a doctor if you are having heart problems. So tissue fluid, we talked about a little bit in the last video, but we're going to link it more to the lymphatic system in this video. So blood contains plasma with water and many other substances like ions, amino acids, hormones, vitamins, plasma proteins. Plasma and some of its components are going to leak out of the capillaries near an arteriole under high pressure to the cells, and this is called tissue fluid. And so you can see here all of the different components that are coming out, but what is not coming out is red blood cells and plasma proteins. But glucose, oxygen, water, amino acids, all of these things are going to come out, and then water, waste, and carbon dioxide, waste would be urea, are going to be flowing around the tissues, the cells. Cells can absorb oxygen as well as glucose and amino acids from the tissue fluid for use in the cell, and CO2, urea, and other metabolic wastes are picked up and moved for removal via the lungs, liver, or kidney. And so this tissue fluid, most of it goes back into near the venules, and it's just going to become plasma and keep moving in the bloodstream. But about 15% of tissue fluid is not reuptaken by capillaries and drains into tubes called lymphatic vessels. And if it doesn't drain, it's going to cause some swelling, which is called edema. Actually, this is a pretty common problem for example, women who've had breast cancer and they've had their breast tissue removed. Then there ends up being quite a lot of lymphatic swelling because the lymphatic vessels are not draining properly. And the way that the lymphatic vessels drain is that they actually move the lymphatic fluid back up to the subclavian vein. The clavicle is your collarbone here. So there's a vein right underneath your subclavian vein here underneath your collarbone, and that it will re-enter your bloodstream at that point. Once tissue fluid has moved into the lymphatic vessels, it's now called lymph. And lymphatic vessels run throughout the body to collect tissue fluid. Then they merge it into two large ducts, which merge with the subclavian vein to return that to the bloodstream at the vena cava. With mammals, we have a double circulatory system, and that's meaning that it's going to pass through the heart 2 times in order to move oxygen sufficiently around the body. And in order to oxygenate the blood, it's going to need to go to the lungs at low pressure. It needs to be at low pressure so that it doesn't blow up your alveoli. And then once it's coming out of the lungs, it would be too low of pressure for other organs. So your kidneys actually need things at pretty high pressure because they need to filter your blood through the glomerulus. And in order for that to happen, it needs to be a high pressure system. And so it's got to return back to the heart for repumping before it circulates out to the body. And in the double circulatory system, the heart is going to keep low and high oxygen blood, so oxygen rich and oxygen poor blood separated. The words systemic circulatory means to the body, whereas pulmonary circuit means to the lungs. Fish, however, are operating at in a different situation because they're in water. And they are able to pump their blood at high pressure because the gills of a fish prevent high pressure from rupturing the capillaries. The water in the gills prevents that from rupturing. Then the water becomes oxygenated at the gills and circulates to the rest of the fish organs. So there's oxygen in the water that's going to go into the gills, then that moves to the rest of the fish organs. And then the blood returns to the heart after dropping off oxygen and picking up carbon dioxide, and it's going to repump out to the gills. So to summarize, the heart has adaptations to pump high pressure blood. Blood pressure and volume change with contractions of the atrium and ventricles of the heart. Double and single circulatory occur due to necessary pressure differences to circulate blood, and tissue fluid moves out of capillaries into surrounding cells, so it can take up oxygen. The cells can take up oxygen and glucose, and that remaining tissue fluid is going to be uptaken by capillaries, or it's going to be drained into the lymphatic vessels, which return lymph eventually to the bloodstream if your lymphatic vessels are working properly. All right, that's it for this video. I hope you were able to understand almost all of it. Thank you for watching.